Today we are going to deal with hybridization phenomenon. So far we have finished about the pollination, types of the pollination, pollinating agents, that is both biotic and abiotic agents, and the compatibility portion. So that is outbreeding devices we have studied. So there is one more concept which is related to pollination. So that is also called as artificial pollination also, or sometimes it is called as artificial hybridization, or it is also called as hybridization technique. Okay, so sometimes it is also called as hybridization technique. What they'll do, so it is a kind of it is a means of controlled pollination. Okay, what they do in this technique, there will be controlled pollination, especially controlled cross pollination. Okay, so already we know what is cross pollination. Okay, the pollination happens between two different flowers of two different plants. Okay, and the produced seeds or the plants which is produced by the cross pollination, those are genetically different, they show genetic variations. Okay, we know the cross pollination takes place with the help of some pollinating agents. Okay, because they are too far, or the pollination is between the two different plants. Okay, so hence they need what agents for their pollination. In the same way, so here we have studied the agents like some biotic and abiotic agents, like natural agents we have studied. So in this we are going to study like how human beings are artificially, so the cross pollination is controlled. Okay, why they do this hybridization technique? Because of what they do, they take just imagine this is plant A and plant B, or species A and B. From the plant A they take pollen grains, okay, and they dust on the plant B stigma. Okay, this is the stigma we know. What they do, okay, some researchers or the one who is performing hybridization, so what they perform, they collect the pollen grains from plant A variety and they dust it on the surface of stigma of plant B. This pollination we call it as cross pollination. Okay, why this is called as controlled? Because of it is not the pollination is not taking place with the help of any biotic agents or abiotic agents like wind, water, birds, insects, okay, or different kinds of animals. So hence it is said to be controlled. It is controlled by the human beings. So hence what they perform artificial cross pollination. Okay, so this technique, how what they form, the formed plants. Okay, when the pollen grains are dusted on the surface of stigma of a plant B variety, they go and they fuse, fertilization takes place, seeds are formed, and those seeds when they germinate, they produce, when they form into a plant, that plant we call it as now hybrid. Okay, hybrid are the one where there is a combination or mixture of characters, not only morphological, genetical characters. Hence cross pollinations allow genetic variations. Okay, so genetic variation, because they have plant A variety character also, plant B variety character also. Understood? Hence they are called as hybridization. So the production of this hybrid artificially, okay, not naturally by the agents, pollinating agents, but here the control, they controlled by human beings. Okay, So hence it is called as one of the controlled cross pollination technique. Okay, so why they do, why they perform this artificial hybridization or controlled pollination? The first main or the only one, okay, the importance to perform this artificial hybridization is okay, the only one advantage or the funda between to perform the hybridization process, that is artificial hybridization, is to improve the crops, okay, to form the varieties. As we know, if the plants continuously, if they undergo what self pollination, what it causes, the inbreeding depression. Okay, we know already what is inbreeding depression. They will lose what, the special characters, the original characters from their beginning generation. Okay, so hence to avoid, we know naturally plant will switch on to the cross pollination. Plants will encourage cross pollination by performing so many techniques. We have studied the devices. What are those devices? Hercogamy, heterostele dichogamy, dicliny, self incompatibility, all these are the devices. Okay, but when we want some desired characters to be present in such plants, or if I want to make a varieties of rice, okay, rice plant, or varieties of rose flower or different flowers, so then what I'm going to perform? I'm going to perform the artificial hybridization to improve the crop varieties. Okay, okay, we'll see how they perform the hybridization process. So hybridization process performed by different steps, few different steps. The first step is called as emasculation. Okay, third one, bagging, or second one bagging, third one crossing, okay, fourth one tagging. So before tagging, there is one more intermediate step, that is rebagging. Okay, you'll get to know what all these things. Okay, okay, so within the emasculation process, what happens? Emasculation means flower buds. So what happens? Emasculation is the process where within the flower, okay, the selected flower or selected plant, the flower, within the flower, what they do, they remove the anther. Okay, because just imagine they have selected one plant, okay, which contains bisexual flowers, okay, which means they contain both stamen and stem. So in such condition, what they do, okay, they perform emasculation, means at the bud stage only, flower bud stage only, what they remove anther, because they don't need the such flowers or such plants anther, because they are going to put the desired character, mixing the character. Okay, so because of this reason, what they do, they remove the stamen part from the flower at the beginning of the bud stage only. Why at the beginning or why at the bud stage? Because okay, so in the bud stage, within the flowers, the stamens are not matured, the anthers are not matured. From the anthers, the pollen grains are not dispersed out. Okay, so before the maturation of anthers only, before the dispersal of pollen grain, dehiscence of anther, they remove the anther from that flower. That step is called as emasculation. Then one more condition. If a plant consists of only unisexual flowers, then what happens? If the flowers are only pistillate, there is no stamen, then this step is skipped because they don't have already the stamen or the anther to remove. So what happens? They skip this step. Understood? Wherever if the scientist or the person who are performing the hybridization phenomenon selected bisexual flowers within the plant, then only this emasculation process will be conducted, because emasculation is typically to remove the anthers. Okay, so just consider now with the example of bisexual flowers only. Okay, so what happens within the flower? Okay, so there is pistil, okay, and these are anthers. What they do? By the emasculation, they remove the anthers at the bud stage. Now the flower is only containing a female reproductive part, remember. Okay, the flower is containing now, after this step, flower is containing only female reproductive part. Okay, next step is bagging. Using some polythene or butter paper or polythene bags, they cover the emasculated flower. Now this flower is called as emasculated. Why? Because of they perform emasculation, because there is no anther here. Okay, now the flower is bagged. Okay, yes, what happens? There will be bagging. That process we call it as bagging. Why bagging? Why there is reason to cover the emasculated flower? The easy reason is, okay, we know by wind or some insects or some birds there will be cross pollination. Okay, so here why we have removed this anther? Because we need to dust the anther from any different variety. Okay, if any other pollinating agent, if they come and dust on this pistil, then there is no use of artificial hybridization. Why? Because of we need a desired character. Okay, from plant A, a character I need within the plant B. So what happens? Plant A is my desired character, that is the anthers. Okay, I am going to dust on the plant B. So for this reason, what they do, to avoid any cross pollination, they bag it with the polythene bag. Okay, next step is crossing. Now the process comes. They remove this bag during crossing performance. Okay, they remove. Okay, this is plant A. Consider this is plant A, where they have performed emasculation and they have done bagging also. Now they came to crossing step. Okay, why there is time for bagging and crossing? Because of we know there will be synchrony. As I said, there will be synchrony. For the pollen grain germination, the stigma should be acceptable. The stigma should be matured. Okay, after, because what we have done during the flower bud stage only, we have removed anther. It means they need to grow, they need to mature, flower need to get matured. So here also same thing happens. During the bagging phenomenon, no, we keep them to get matured. What? Stigma. Once the stigma get matured, what we do? We perform crossing step. So what is the crossing step? From a plant B, we collect pollen grains. Okay, from plant B, we collect pollen grains and they dusted on the surface of stigma. These pollen grains are dusted. We allow pollination, controlled pollination, that is cross pollination itself, because this is not taking place within themselves. So there is no self pollination, right? From different plants to the different flowers, pollination is taking place. So hence it is called as cross pollination. So crossing means performing the cross pollination. What we have done now, our desired character is a plant A. I need plant B character in the plant A. So what I'll do now, I'll take the anthers from the plant B, different plant, and I dust on the surface of the stigma. Okay, this is, we performed what? Cross pollination. Okay, we can write this as a cross pollination. Okay, that too controlled one. because we are performing, humans are performing, not by any agents. Okay. Next, what we do? The process is rebagging. Again we do rebagging. Okay, same thing to avoid the transfer of other different plants pollen grains on the surface of stigma. To avoid that, what we do, again we rebag and we keep it for next step, that is fertilization. Okay, the pollen grain which is dusted on the surface of stigma, they get matured, they get germinate, and they fuse with the egg of that stigma. What happens? Then seed is formed, fruit is formed. Okay, with the help of seeds, seeds will germinate when we sow that seeds, and the hybrid is formed. That hybrid now contains the mixture of character. Plant A is a character and plant B is character. So this is what artificial hybridization. And simple thing, the tagging is nothing but what we do. Okay, we make some tags and we tag that plant. Okay, writing the name of the original A plant. Okay, the name of the original B plant, from which plant we have taken pollen grains, which day we have emasculated, which day we have performed cross pollination. Okay, and person, it may be from which institution of which person. This is called as a tagging. Okay, this is simple thing to know. Yes, we have done or we have performed the pollination, artificial pollination or artificial hybridization to this plant. To indicate this, we are going to tag it. Okay, so this is what we call it as artificial hybridization. So next we'll study the pollen pistil interaction, and we'll go to the next, that is fertilization event. This ends with the pre-fertilization event in angiosperms.